When we were together last, we looked at this diagram which provides a summary of the two sets of reactions involved in photosynthesis. Remember that it's sunlight that drives this process, sunlight which provides the energy, the light energy that's captured during the light-dependent reactions and used to produce two chemical molecules, ATP and NADPH. So yesterday we talked about how that happens, how photons interact with pigment inside these photosynthetic organisms, inside the chloroplasts and the thylakoid membrane, and excite electrons in order to... The last time we were together, we looked at this diagram, which provides a very general summary of photosynthesis. Starting at the top, it's the energy of the sun that provides the electrons to drive this reaction forward. We talked about how the light-dependent reactions, the ones we discussed yesterday in class, produce two important chemical intermediates, ATP and NADPH. And it's these two molecules that are going to help drive the remaining set of reactions of photosynthesis called the Calvin cycle. Remember, we said that a photosystem is just a group of chlorophyll molecules and some accessory pigment. 200 to 300 of these molecules clustered together inside the thylakoid membrane of the chloroplast. We said that photons hit the chlorophyll molecules and get passed around the outer molecules, which are known as the antenna complex. When this energy eventually reaches the chlorophyll molecules in the center of the photosystem, the reaction center, we see the actual electron lost by the chlorophyll molecule and gained by something else. So the chlorophyll molecule in the reaction center gets oxidized, and then something else gets reduced. And in the case of photosystem 2, that something else is a molecule called phyophytin. So if we put it together, that means that the absorption of light causes the reduction of phyophytin and the oxidation of chlorophyll. It's the phyophytin molecule that can then donate electrons into the electron transport chain, which again is located in the thylakoid membrane, just like we saw the electron transport chain situated in the inner mitochondrial membrane. The two are similar, but they have some important differences. Remember, in the mitochondria, during cellular respiration, we had these complexes of proteins that helped step down the electrons through the electron transport chain. We said these proteins were unusual in that they had very strange side groups, things like iron and sulfur and heme. Well, the electron transport chain in plants is very similar. It's got some unusual compounds in it that are also capable of stepping down electrons in energy. But the other important player we mentioned in the mitochondria was ubiquinone. We said ubiquinone was a quinone molecule that was highly hydrophobic, lipid soluble, so it was capable of traveling around the tail region of the inner mitochondrial membrane, and actually performed two jobs. It helped transfer electrons from protein to protein to protein in the electron transport chain, and it also helped pump hydrogen ions into the intermembrane space. Inside a photosynthetic organism, we're not going to find any ubiquinone, but there is a very similar compound known as plastoquinone. Plastoquinone is another quinone molecule, another molecule that's highly mobile within the photosynthetic thylakoid membrane. The electron transport chain inside photosynthetic molecules also contains cytochromes. And we saw these in the mitochondria as well. Remember, cytochrome C was capable of helping ubiquinone in the process of transferring electrons down the electron transport chain. Same things happening in a photosynthetic organism. We have the plastoquinone helping to transfer electrons down the chain, and we have cytochromes as sort of assistant molecules. And in the process of electrons getting transferred down the chain, protons are going to be pumped across the membrane and into the lumen of the chloroplast. And just as we saw in the mitochondria, it's this proton gradient that will drive the production of ATP through a molecule called ATP synthase. We've seen something very, very similar to this in the mitochondria. This is a very efficient way to make ATP. The big difference is that in the mitochondria, the ATP produced was going to be used by the organism for general energy needs. But as we said in class, the ATP that's produced by photosystem 2 is going to be immediately used as a substrate for the Calvin cycle. Now, notice on this slide there's a term that we haven't seen before, a couple of terms actually. One is a proton motive force. Anytime we have a proton gradient and the force of that gradient, the potential energy of that gradient is used to do something else, we can call that a proton motive force. The other term you see that's new for us is chemiosmosis. Chemiosmosis can be defined as coupling a proton motive force, a proton gradient, with the production of ATP. We see this both in cellular respiration and in photosynthesis. We see a proton gradient flowing through ATP synthase and being used to make ATP. Now this slide provides a diagram for us of photosystem 2 as it sits inside the thylakoid membrane. So you can see the section of membrane that we're being shown. You can see the phospholipids in a bilayer. This space in here represents the lumen, and this space out here represents the stroma inside the chloroplast organelle. Now, a couple of things to notice. Here's photosystem 2. Here are those 200 to 300 chlorophyll molecules all grouped together to work as a unit. And here is the electron transport chain. Unlike what we see in mitochondria, in the chloroplast, all of the proteins in the electron transport chain are grouped into a single complex, and that's referred to as the cytochrome complex. If you go back over to the left-hand side of the slide, you can see the incoming photon. It's striking some pigment in the antenna complex. Remember, that energy is going to excite the electron, and then as the electron drops back down to zero state, the energy will be transferred from chlorophyll to chlorophyll to chlorophyll around the antenna complex until ultimately the energy reaches the reaction center. When the electron in the reaction center gets excited, it doesn't drop back down. It gets lost. It gets transferred to pheophytin. You can see that right here. Pheophytin will then transfer the electron into the electron transport chain with the help of the quinone called, again, plastoquinone. And it's plastoquinone that will help move the electron down the electron transport chain, releasing small amounts of energy with each step. Just like ubiquinone did in the mitochondria, plastoquinone can also pump hydrogen ions. It can pump protons and help build up this gradient inside the thylakoid lumen. Ultimately, we have our old friend ATP synthase. Now, in this drawing, it's shown oriented in the other direction compared to the drawing we saw in the mitochondria, but it's the same molecule. It has a portion embedded in the thylakoid membrane, it has a stalk, and then it has the portion that spins. As the protons that build up inside the lumen, as they develop this gradient, they will ultimately flow through this subunit of ATP synthase and cause this one to spin and change shape, which provides the force to make ADP into ATP. So, plastoquinone is fully capable of moving around inside the thylakoid membrane. It receives electrons from pheophytin, and of course protons follow. Usually when a molecule receives an electron, it also receives a hydrogen atom. It carries the electrons across the membrane and delivers them to the next protein in the electron transport chain. Remember that all of the electron transport chain, electron acceptor molecules, the proteins, are found in this big complex called the cytochrome complex. And just like we saw in the mitochondria, these proteins are a little odd in that they're associated with things like iron and copper and heme. The other job that plastoquinone does is it carries protons from one side of the membrane to the other, helping to build a proton gradient inside the thylakoid lumen. So here's our plastoquinone doing two different jobs, both helping to transport electrons through the electron transport chain and helping to build a proton gradient. You'll notice that there are very few hydrogen ions over here, lots and lots of hydrogen ions inside the thylakoid lumen, and when they are allowed to flow through the ATP synthase molecule, that's how ATP will ultimately be produced. So, pheophytin donates electrons to the electron transport chain inside chloroplasts, but we see some real similarities between what happens with ATP synthase in the mitochondria during cellular respiration and what happens in photosynthesis. In both of these locations, protons will diffuse down the gradient and drive the synthesis of ATP by allowing the ATP synthase molecule to change shape. That conformational change in the ATP synthase helps phosphorylate ADP to become ATP. Now, something important to note down here on the bottom of the slide is that plants have a lot going on in terms of the fact that they perform both cellular respiration in their mitochondria and they perform photosynthesis inside their chloroplasts. So chemiosmosis, the use of a proton motive force to produce ATP, is occurring both in the mitochondria and in the chloroplast in plants. So in photosystem 2, what we ultimately see occurring is that light energy is being captured by chlorophyll and then converted to chemical energy in the form of ATP. And because we used ATP synthase to accomplish this, this is similar to oxidative phosphorylation, but it's a little bit different. We didn't see an oxygen as the ultimate electron acceptor at the end of the electron transport chain. So instead, this kind of ATP production is called photophosphorylation. Photo meaning light phosphorylation. So the phosphorylation ultimately is energized with light from the sun. So we now know three ways that a cell can make ATP. We know about substrate level phosphorylation, the kind that we saw during glycolysis and during the Krebs cycle. We know about oxidative phosphorylation, which is what's happening in the mitochondria during oxidative phosphorylation. And we know photophosphorylation, which is occurring in the chloroplast and helping to produce ATP. So here's a final question. The electron that was transferred from the reaction center in the chlorophyll to the pheophytin molecule was lost, and the pheophytin was oxidized. Now, we have to replace this electron. We have to replace the electron in order for photosystem 2 to continue to operate. In other words, we've got to reduce the reaction center again so that there's an electron in the reaction center that's ready to be excited when more photon energy is absorbed. We know that electrons come from the sun, energy comes from the sun and through the photon, but we can replace the phyophyton electron through a process using water. And if you recall, when we looked at the summary equation for photosynthesis, we said carbon dioxide and water and light energy are used to produce carbohydrate, glucose, plus oxygen, plus water. So once again, we have oxygen here being produced, but it's being produced from water. This is an unusual way to make oxygen. This is what we refer to as the splitting of water. So water is taken apart to produce oxygen plus electrons plus hydrogen ions. Remember, when we looked at the mitochondria and cellular respiration, we saw something similar going on, but it was really in the reverse. So at the end of photosystem 2, we've produced ATP, we've provided electrons to reduce our to reduce our reaction center once again. But at the end of the electron transport chain, we now have an electron that needs to be accepted by another molecule. And the question is, what is that final electron acceptor? And the answer might surprise you. The answer is it's the next photosystem. It's photosystem 1. If you remember in class, we talked about how photosystem 1 was actually the first photosystem that was investigated and explained in terms of its physiology, which is why it was called number 1. It was identified in bacterial species. And what the early scientists learned was that instead of passing electrons to the electron transport chain to pump protons and make ATP, in photosystem 1, the electrons are used instead to reduce an electron carrier molecule. Remember, in photosynthesis, this electron carrier molecule is a little bit different. It's not NAD, the molecule that we saw previously in mitochondria and cellular respiration. Instead, it's NADP+. Now, there are still some bacteria on the planet who use NAD as their electron carrier, but most of the cyanobacteria, the algae, and the green plants on the planet use this molecule, NADP+. And when it accepts electrons, when it gains electrons and gets reduced, it becomes NADPH.